Hello, everyone, and welcome to today's interactive program. My name is Sarah Crown-Starr, and I'm the president of SCS Innovations, a Chicago-based investment firm. I'm thrilled to be hosting this event on behalf of the Rappaport Technion Integrated Cancer Center, exclusively for donors to the center. For many years, I and my family have been strong supporters of Technion. 5 years ago, we established the Crown Family Histopathology Corps and Faculty Recruitment Fund in order to attract the best and brightest researchers to the Cancer Center. It is a privilege to be able to play a role in furthering the state of our knowledge of cancer treatment and prevention in this way. Without further ado, let's jump right into today's event. In a minute, I'll introduce Professor Yuval Shaked, the director of the Rappaport Technion Integrated Cancer Center and a member of the Department of Cell Biology and Cancer Science at the Technion Rappaport Faculty of Medicine. We will all then hear from each of today's three amazing speakers, I might add. Professor Irit, Ben-Aharon, Associate Professor Avi Schroeder, who is one of the inaugural recipients of the Crown Vanguard Award for Science and Technology Innovation, and Associate Professor Asya roles. You are all muted now, but a moderator will unmute you during the Q&A period. For the best viewing experience, please go to speaker view. And if you're unsure of how to do that, just hit the chat button down below and someone will help you. Welcome, and it's now my pleasure to introduce Professor Yuval Shaked. Yuval, please go ahead.
Thank you very much. 1 second. Yeah. So thank you, Sarah. Hello, everybody. I would like to start by offering my sincere thanks to each and every one of you who has supported and who continues to support and will support the activities of the Rappaport Technion Integrated Cancer Center. You're all aware of the importance of the work being conducted at the frontiers of cancer research today. It is my honor and privilege to head an institute that is playing a world-leading role in this quest. What you'll see on the screen is the design of the new building of the Rappaport Technion Integrated Cancer Center. The RTICC was founded in 2015. It's focused on discovering new diagnostic tools and treatments. The interaction of researchers in all areas of science with engineers and clinicians, including oncologists, helps us translate basic discovery into medical applications. This process culminates in clinical trials at our 5 affiliated hospitals in Israel and beyond, as I will show you later on. Over the years, the center has trained graduate students, postdoctoral fellows, clinical fellows, and visiting students and scientists. And as you can see here, the design of the building is a beyond state-of-the-art infrastructure within this building that will put together both engineers and clinicians, along with basic researchers, all of whom will talk in the same language, the language of cancer research. The RTICC integrates efforts between engineers and basic clinical experts in order to address unmet needs in clinical oncology. The engineer arm is the arm that will work on drug delivery, on drug design, system biology, computational science. Professor Schroeder probably will talk at some of these aspects. Basic and translational cancer research, where we look at tumor dormancy, why tumors start to metastasize, the resistance to treatment, metabolism. I will touch base on some of these aspects. And clinical research and clinical trials, dealing with personalized medicine, biomarkers, new treatment modalities, new treatment approaches, discussing by our Irit Ben-Aharon, Professor Irit Ben-Aharon, and Professor Asya. The goals of the RTICC is to enable paradigm shift in cancer diagnosis and therapy, is to bridge between engineers and biomedical faculties. I think what the unique about the Technion is the fact that we use the engineers in order to advance cancer research. It's to enable bedside to bench and back to bedside. These interactions and collaborations are important for synergy of a clinical outcome, and I will show you some of these aspects in my own presentation, and to establish a personalized medicine practice in the north of Israel. And when I say personalized medicine practice, I would like to move into my own research at the RTICC. One of the major goals of the NIH-NCI is to have the initiative of the precision medicine in oncology, is to develop this in order to improve the life of many cancer patients. And we set as a goal in my own lab to study more the precision medicine in oncology. As you may know, the tumor mass consists of many different cell types. These are not only tumor cells, but also additional cells within the tumor mass. Each and one of these cells will support the growth of the tumor and will promote metastasis. In my own lab, what we study is how therapy affects this tumor microenvironment. When the majority of studies around the world are focusing on how therapy target tumor cells, we actually look how therapy affects the body and how the body reacts to the therapy. This reaction, as we have learned, is changing the way tumor cells behave, sometimes to the tumor advantage. So the forces between the action of the therapy on the tumor and the reaction of the host to the treatment change the tumor microenvironment and make tumors sometimes regrow and metastasize. I want to give you an example how it looks like in real life. Here what you can see are tumor cells. When I take blood from a lung cancer patient before treatment and put on these cells, I can ask how these cells behave when they meet the blood, the plasma of the patient. I can take from the same patient the blood after treatment, and I can ask how the cells now behave when they meet the blood of the patient after treatment. And as you will note, there is a huge difference between the blood before treatment and the blood after treatment. The blood before treatment, what you will see is the tumor cells slowly close the gap. That means they are less active. Whereas after treatment, you can see that the tumor cells are more aggressive, more excited, and actually close the gap much faster, which means they are more protumorgenic. They generate more protumorgenic activities and can metastasize. How can we use that in order to improve life of cancer patients? That's the question. In the last 3 years, I founded a company called OncoHost, which works on precision medicine and oncology. It was founded based on the work performed at the RTICC. What we do, we take blood samples from patients before treatment and on treatment. We look at the different factors in the plasma that can explain regrowth of tumors, the factors that can promote the growth of tumors. Based on this, we used a bioinformatic analysis and machine learning technique. I call it RTICC, in order to stratify between those that respond to the treatment and those that do not. And then we generate a response prediction report, and maybe also can suggest other treatments that will be of benefit for these patients. Here is an example of a report of a patient with non-small cell lung cancer treated with immunotherapy. We predicted that the patient will not respond to the treatment, and indeed, within 3 months, the patient succumbed to the disease. Currently, there is a clinical trial that Rambam Hospital and Professor Irit Ben-Aharon are tightly involved in this clinical trial called PROPHET, in order to predict outcome of patients following treatment with immunotherapy. As you can see, there are many, about 12 centers in Israel that are involved in this trial, as well as centers in North America, in America, in the US, such as Fox Chase, Yale Cancer Centers, Sindel Kimmel Cancer Centers, and others that will come aboard soon. In addition, we also have cancer centers that will start in the European Union. If you recall, 2 years ago, I have met probably some of you, and we discussed that we will launch a clinical trial, and here it is. This is the clinical trial, and it's all based on the commercialization of the studies, the basic research done at the RTICC, and become a real thing for the benefit of patients. And with this, I would like to thank you for attention, and I would like now it is my pleasure to introduce now our next speaker, Professor Irit Ben-Aharon, the Director of the Division of Oncology at Rambam Healthcare Campus. Irit, the stage is yours.
Hello, it's my pleasure to be with you. It's my honor, and thank you for your support. Unfortunately, it's in this Zoom format. I wish it was not. I would like to take this opportunity and tell you about not only my own research, which emphasizes on the toxicity mechanisms of anti-cancer treatments, but rather on something that is very up to date, which is the COVID-19 and the potential impact on cancer patients. And this was actually a very natural and classical collaborative project between the TICC and Rambam, because my just natural partner was Professor Yuval. He was the first one that I approached when I thought maybe we should think of another aspect rather than just having the assumption of cancer is high risk for COVID, and maybe we should explore other possibilities. So I would like to share with you this work. I would just mention a little bit about, is it working? Yeah. About Rambam. Rambam is the largest cancer center, the largest tertiary hospital in the northern part of Israel, and the largest cancer center in this part. And we actually serve 2.3 million residents. So we have very high capacity of almost 5,000 new patients per year. We are the largest radiation oncology facility, and we have many clinical trials, pharma, and also translational research that we have in our facility. This project or the research that I want to share with you is actually something that we thought we came with this idea at the beginning of the pandemic, when despite the lack of concrete evidence, cancer patients were considered as a high-risk population, like they usually are considered, where we talk about infectious diseases. And previous reports on the incidence of COVID-19, at least at the beginning of the pandemic, came from China and Italy, and they were consisted of retrospective studies. And the data was very conflicting. In some of those studies, the incidence appeared to be high. In some of them, it was the same. So when those studies appraised the clinical course and mortality in symptomatic COVID-19 patients, we saw that the comorbidities that we know today that are considered as high risk factors for COVID, for severe course of COVID-19, were hypertension in older age, and also in cancer patients. In some of those studies, these were the key factors to determine the clinical course rather than the cancer or the anti-cancer treatments themselves. But despite this conflicting evidence that we had, cancer care had been revisited while postponing chemotherapy or advising to postpone elective surgery for specific indications in endemic areas. It was worldwide. And some of those association that revisited the guidelines had no evidence to generate those guidelines. So we sought to really explore whether cancer patients are indeed at high risk, and we wanted to study that prospectively. And we thought that the best control would be the oncology healthcare workers at Rambam, because they had the same exposure. Everybody stayed at home and they just came to work or to receive the treatments for the patients. And we wanted to also explore the immune profile because the assumption was that maybe the immune profile that is different in cancer patients may interfere with the induction of the cytokine storm that is typical for this disease. So we wanted to have this immune profiling of those infected ones. So what we did is actually we started in March, the beginning of the pandemic in Israel. For 3 months, the infection rate in Israel the incidence was very low in that period of time. What we did was that prospectively, we just enrolled all the patients that agreed to be enrolled into the study, and the ones that came to treatment, and also from the healthcare workers. We sought to explore the expression of the antibodies at several time points, and in collaboration with Yuval, we performed the immune profiling at the RTICC. So we had 164 cancer patients that had the active IV treatment for their cancer, and 107 healthcare workers at our center. And this is actually, if we want to describe the time frame, you can see here that this was between March and June, and half of this period was the first lockdown in Israel. And what we actually saw that there were no symptomatic cases throughout this period. And I have to say that today, 8 months later, we have very few cases, although the pandemic became much more prevalent in Israel later on. And the serology analysis of that time revealed that the rate of infected patients, all of them asymptomatic, was the same between the cancer patients and the healthcare workers. And we had only 4 cases of patients that we had in this cohort that were IgG positive, and most of them received chemotherapy, and some of them received immune therapy. So what we did is actually went to profile their immune cells, the white cells, and what we discovered is that in the healthcare workers, the CD8 is a typical white cell is different, if you can see here, between the healthcare workers that were exposed to COVID and the ones that were not. And in the cancer patients, there was no difference. And then if we go to the myeloid cells, it's another cell type. There was a drop in the healthcare workers that were COVID positive, and there was a drop also in the cancer patients, but it was much more moderate than the one that we saw in the healthcare workers. So if we want to go for the mechanism, the putative mechanism that we think underlie what we see here, is that previous studies indicate that cancer patients undergo, as Yuval showed, immunological alterations toward the anti-inflammation state due to the treatments. And what we see, limited by the small sample size, we now have this study ongoing for the larger sample size. It may imply the differential immune profile for the cancer patients when we compare it with the healthy subject. And what we know of on the COVID is that it induces the cytokine storm. And our hypothesis is that the anti-cancer treatment that induced the modification of the immune profiling may blunt this cytokine storm, and therefore it's not that they may not be infected by the virus, but they would not generate this very severe pattern of the disease. So what we can conclude is that we saw that the infection rate is the same, and due to the differential immune cell profile, we may think that the host response may play a role in the COVID course and the representation in cancer patients. And the immunological perspective of cancer treatment on the risk of COVID-19 infection, of course, should be further explored. This is only preliminary study, but nowadays, since we have the pandemic got much worse in Israel later on, this study was concluded in June. We continue and we hope that we will be able to reveal some of these mechanisms. Thank you. I am now very pleased to turn the program over to my colleague, Professor Avi Schroeder, who heads the Lewis Family Laboratory for Targeted Drug Delivery and Personalized Medicine Technologies in the Faculty of Chemical Engineering.
Thank you, Irit. Thank you so much. I'll share my screen with you. And the case I'd want to make here tonight, and I think Yuval and Irit and Asya are going to make it even stronger than I can, is that the Technion is all about collaborations. It's about collaborations between medical doctors and engineers and basic scientists. And it's also a lot about collaborations with you, our friends from across the sea. And we all really, as Irit mentioned, we all would want to be with you in the same hall tonight, but we're here, and I hope in a couple of months we can actually see each other. And really, the case I would want to be discussing tonight is actually nanotechnology, and we all heard the news this morning, and it's again a nanotechnology news. So the recent medicines that are being used, or the vaccines that are being used for treating COVID, a lot of them are nanotechnology based. On the left-hand side, you can see the Moderna nanoparticle. This is a nanoparticle, which is very, very similar to the nanoparticle that Pfizer is talking about. Its size is about 1/1000th the width on a hair on our head. And it actually encodes inside it special RNA molecules or messenger RNA molecules that encode the production of proteins inside the body that are associated with COVID. And that's how our immune system actually detects them. And that's how a vaccine is actually caused inside our body. So nanotechnology is actually out there. It's being used in cancer therapy. It's being used throughout the medical field. And COVID is one of those examples. But I would want to go back and actually talk about cancer. Specifically, one of the main hurdles that we have in cancer, and this just emphasizes it. This just before the COVID broke out, I was invited to give a talk in a medical conference, and I was the scientist there, and they were all medical doctors around, and they asked questions. These are clinical questions. Irit, I'm sure, can answer it immediately. And I was astonished. So they give the situation. It's a female patient, and she has breast cancer. It's node-negative breast cancer. And then they go on with the biological descriptors of her specific tumor, and they ask the physicians, how should we treat her? And being an engineer, I said, oh, well, I'm sure that, one of the answers will get most of the votes. And the votes were cast, and this is actually the screenshot that I saw, and this was the screenshot. It varied. These were 350 medical doctors in the audience. It was really hard to decide in this specific case how to treat this specific patient. And I think we're all aware of it. We hear sometimes of a patient that responds to medication. That's great. We're all happy. And in other cases, there's no response at all. And we asked ourselves, could we use nanotechnology to help predict which medication will be best for treating each cancer patient? And what we did is we created a system. They're called barcoded nanoparticles because they're very much like barcodes we have in the supermarket, but here it's actually in these extremely small particles, and each one of these particles carries a minuscule dose of medicine. Each nanoparticle, a different medicine. And the nanoparticles are engineered in a special way so that after being injected intravenously, they actually target the tumor site and give us a readout about which medication works and which medication doesn't work. So by carrying different medicines inside of them, we can have better understanding within a very short period of time which medicine is best for treating each and every patient. In fact, today the science goes even a bit deeper. Irit spoke about different cell types that we have inside, and Yuval emphasized that some cells respond to one medicine while others respond to others. We actually today, using this technology, we can predict which medicine would be best for treating a primary tumor, the main local tumor, and which medicine would be best for treating the metastasis in other locations, and what medicine should we not treat our patients with because we may actually face horrible side effects. And what this technology at the end of the day does is for each patient, it creates a potency chart, a chart that says this medication is best for treating this specific patient, while let's not choose another medicine for treating that specific patient. And it's all based on that we all are unique. The personalized medicine theme that Sarah actually started with, we need to treat each patient according to many, many different factors that exist inside their body. And it proved pretty well. This is still in preclinical trial. We saw that we were able to shrink the tumors better. And also the animals that were treated according to the prediction lived far longer. And I think just to explain how this technology worked, this small animation can actually give a nice picture. So these are the barcoded nanoparticles injected into the blood. They're engineered so they penetrate into small permeation sites inside the tumor. And inside the tumor itself, they actually can distribute different medications, giving us the readout about which medicine worked and which medicine didn't work. But we had a problem when we were developing this technology. And we weren't able to actually differentiate between the live cells being taken out of a tumor and the dead cells inside the tumor. And that's where we met Professor Yuval Shaked, who's on the call tonight with you. And he actually gave us a lot of great tips and a lot of lab help about looking at the different cells inside a tumor. But he also, as I was walking out of the room, and we already had this technology and it's actually being translated as we speak to the clinic, he said, well, Avi, you know, breast cancer, which you guys are working on, has several technologies, is extremely important and affects so many lives. But also pancreatic cancer is an extremely important medical target. But in pancreatic cancer, there's another problem. There's actually a very, very stiff stroma that stops medicines from penetrating into the tumor. Maybe your nanotechnologies can help with penetrating tumors a little better. And when we made histological cuts of the pancreatic tumors, we actually saw lots of this blue material inside. It's not really blue. This is a blue stain that we use with a chemical called Mason trichrome, and it stains the amount of collagen we have inside. Collagen is fibrotic tissue. It's a little bit like scar tissue directly inside the diseased pancreas. And this scar tissue prevents medicines from penetrating into the pancreas. And what we did, actually Asaf Zinger started working on that. Asaf is a former student of ours. He just is finishing his postdoc in the US and signed the Technion 2 weeks ago to return back as faculty. He invented nanoparticles that carry inside them a specific enzyme that degrades the extracellular matrix inside the tumor. So you actually drill small holes into the pancreatic tumors, allowing medicines to be far, far more effective. But one time, Asaf was down in the MRI in the Rambam Hospital. Our lab is located on the mountain. He was sitting there with Maria. Maria is now a graduate student in our group. At the time, she was an undergraduate student. And they were looking at a very strange biodistribution problem. So he calls me up and he says, Avi, listen, you have to look at this picture. And it was like the end of the day, 7 in the evening. And I say, okay, I wait. He came up and he shows me this. And I said, Asaf, this is what you kept me held up for? And he said, yeah, don't you see? I said, yeah, it's in the urinary tract. All the medicine all this expensive medicine is actually being washed out of the body. He said, no. And I said, yes. And of course, he was right, as students usually are. And what happened actually is that these are female mice, and the nanoparticles were accumulating in the ovaries of these female mice. Now, in our group, we had no knowledge of ovaries and how nanotechnologies should actually work inside the female reproductive system. We started reading all about it, but then we said, you know, let's go to the #1 expert in the country and probably one of the best in the world in this field, Professor Irit Ben-Aharon, who's also on this call tonight. And we started asking her, Irit, what would happen? Does that even make sense that nanotechnology can reach the ovaries, can reach the uterus during ovulation? And we were looking at the technology, and actually it all made sense. And to make a long story short, is from the barcoding system, which started a lot with a collaboration with Yuval through a collaboration with Irit Ben-Aharon, today we can actually leverage the biodistribution of nanoparticles to different regions inside the body for treating different conditions. In some cases, you want the nanoparticles to reach the ovaries, such as when there is ovarian cancer, ovarian tumors. And in other cases, you don't want the nanoparticles to reach the ovaries because then they can actually harm the fertility of a person. But it did open a whole new technological field. So you can see Maria on the right-hand side of and Asaf right next to her. And they actually started a new field, which is gender nanotechnology. And it's how actually our sex relates to how different nanoparticles work inside our body. So the Technion is about is a lot about collaboration, and it's about looking at using different tools to look at where the nanoparticles, where the medicines reach inside our body, leveraging that for improving therapy, such as breast cancer, pancreatic cancer, and in certain cases also ovarian cancer. But I'd like to end with where we are today in our laboratory. And that's really Professor Asya Rose, who will be the next speaker right after me. And so today we understand that it's not that we have our brain and that we have our body. It's actually the two are connected. It took me several years to understand that. My wife says too many years to understand that. And many times our brain will regulate how different disease will occur inside our body, and we can't differentiate between the two. And this is an exciting project. It's still really, really early on. So I hope if you guys allow us, we'll be able to present it later on, but I will pass it on now to the exciting research that Asya Rose is doing in her laboratory. Thank you so much, and thank you for supporting the Technion.
Thank you, Avi. It's been a pleasure. I was fascinated here. So, and it's like, it was like a treasure story or treasure hunt story. It's always fun. And I think it does reflect on how science is, right? It's like we are always, the more confused we are on the right path. So I think it's definitely sounds like it. And indeed, we're now working on I think we spent many hours this afternoon talking about new projects that we are working on together with Avi. So it's fun meeting later on with everyone. And as Avi said, it is a collaboration with everyone. And seeing just the faces when you got into the room, I realized that we actually know a lot of you, and we've met over the years, and it's almost a family-like atmosphere already. So I will tell you about our stuff. I will put it a little bit in a different context and what's going on these days. I think I will share my screen. What's going on these days, and I think these are interesting times that affect many aspects of our being, and it's not only the emotional, it's not only the physical condition that is being changed with this COVID and the threat of the virus. It's a lot of it. It's also our mental state. And I think so many things have changed in the last year. And the feeling we are more isolated. We are a lot of stress that came into our lives. And actually, this is something that has been connected over the years with the emergence of cancer and increased susceptibility to cancer when stress is there. One of the reasons and one of the explanations is that it is because when we are stressed, there is a suppression of the immune system, which is supposed to fight the tumors. And that this suppression is kind of allows the tumors to emerge, and because they're less controlled. But what I want to tell you is that it's not just the stress, because stress is something that is built in our body. It's essential for us to exist. If our stress systems do not work, we cannot even wake up in the morning. We cannot get out of bed. It's essential for us. Like when the stress systems are not functional, many of the other physiological systems don't work. So it's about being synchronized, and it's about the balance, because sometimes stress can even improve immune activity. And part of it is how our attitude to stress and our attitude to different aspects of our physiology. And I want to give you an example from a very interesting study that was done many years ago in Yale by Alia Krum. What she did, she gave people milkshakes. One was kind of an indulgent milkshake, and the other was a skinny milkshake. And she asked people to taste the milkshake. They thought that they are participating in a study, for a dietary study, and they collected blood samples. And they followed in the blood the levels of a hormone called ghrelin, which is the hunger hormone. And as you can expect, in people that got the indulgent milkshake, they had lower levels of this hunger hormone, and those who got the skinny milkshake actually had higher levels. The thing was that it was exactly the same milkshake. So what was different is what people thought about the milkshake. So it kind of speaks to how it's not only what we eat, it's how we think about what we eat, and our hormonal system levels will change accordingly. We adapt things. And even exercise. So we think that it's, yes, of course, it's important to exercise, but how we think about exercise also matters. So for example, what Alia Krum, what she did, she said, okay, let's take people who are not exercising. They don't think they're exercising, but actually they do. And let's see if just by changing the attitude, we are going to change something in their physiology. So they took hotel mates. And these hotel mates, actually, they work a lot. They do a lot of exercise, but they're not aware of it. They're treating it as work. So they gave them a workshop and to this hotel mate, and they explained to them that what they're doing is exercise, and they should benefit all the benefits of exercise, changes in their heart rate, changes in their cholesterol levels, even changes in their waistline. So, and then they compared, and the other group was just got like a explanation for why it is important to get exercise. And then they compared it, and after a month, they realized that the women that thought that what they are doing, their work is actually exercise, got much better. They actually got all the physiological benefits of the exercise themselves. So, and it goes on. If you, for example, take people, and it was a big study, several studies actually that were done in people that are getting medications to relieve pain. So they had people who got pain relievers after surgery, either open injections, so the nurse would come and say, okay, now I'm turning on the painkillers, and the other group will just get it hiddenly, so the computer will turn on the delivery of the drug, got at the same times, at the same doses, everything is the same. But have a look here. This is the outcome of pain relief. So you see here the reduction in pain. In gray is those who got the open application, so they knew that they are getting the pain reliever. And those who got in the orange, you see that it's much less for those who didn't know they were getting it. It was just turned on by the machine. So we're talking here about a pharmacological effect, and this is a psychological effect, which seems to be much stronger and almost kind of unused in medicine. And of course, we know that social support and positive attitudes seem to be correlated with improved outcome from different diseases, including cancer. And the placebo, which has been there for 50 years now, that we are giving people a sham treatment, and they seem to get better in some cases, even just from the treatment. And the thing with placebo, and I want to give you an example from recent FDA case, and I think FDA is in the news these days, so I just want to show you. One of the things that is happening is that the efficiency of the placebo group is actually rising. And this is an example in pain medications. So in 96, when they compare different treatments, it is a summary of 35 clinical trials. The difference between the treatment group and the placebo was about 30% better in the treatment group. So for those who got the medication compared to those who got some sham treatment. In 2013, it was actually only 9% difference. So you ask yourself, so what did we get so much worse in creating new drugs? Well, apparently the thing is that people in the placebo group just got better just from getting the drug. And it seems, and this is interesting, it seems to be mainly in the US, actually. And the bigger the clinical trial, the more, the higher the placebo response. And you know what the worst thing for a clinical trial, what will increase the placebo response dramatically? If there is a dedicated nurse. If there is a nurse who actually will ask you, how are you doing? And will give you administer the drug, that's the worst. So there is some sort of disconnect there. We see this effect of our physiology or our thoughts and emotions, psychological state on the clinic, but we don't know what's going on. It seems to be disconnected in the Descartesian actually way. And what we are trying to do is to understand how it's connected. And we are doing this connection by saying, okay, so thoughts and emotions, they affect specific brain activity. In placebo, it's this positive expectation to get better. And it's activating a certain brain area, the reward system. So what we are doing, we are asking how activation of the brain's reward system will affect the body's ability to cope with disease. And this was done, this was work of Tamar Ben-Shaanan in the lab. She's now a postdoc in UCSF. And another member of the RTICC, Shai Shenor, who is the systems immunologist. And what we ask is, if we are going to manipulate the reward system of mice, this area that is activated when there is positive expectations, are we going to change the mice immune response? And we can do that by precisely manipulating specific neurons in the brain. And that's what happened. We activated the brains of the reward system of mice, extracted them from their blood immune cells that usually kill bacteria. So we incubated them with bacteria and then plated the remaining media here. So you see every dot represents the amount of bacteria that is remaining. And you see that there are much less dots after reward system activation, suggesting that just by activating the brain, the immune cells of these mice were more potent in killing the bacteria. And we also said, okay, because the immune system is so important also in killing tumor, can we manipulate by using the brain, the immune system of mice? And this was again the work of Tamar, another MD PhD student in the lab, Maya Schiller, together with Dr. Faid Hakim, who is the head of the Nazareth Medical Institute, and Professor Mikirat, she's also a member here in the Technion. And what we did, we activated the reward system of mice that were injected with tumor cells. And you can see that this is the size of a tumor in control mice, and this is the size of the tumor in mice that we activated the reward system. So, well, it's only one case, but no, you can see that it happens in different mice. And every time when we are manipulating their brain, we can reduce the size of the tumor. But how does it happen? We are manipulating the brain. How does it change eventually the immune response in the body, somewhere in the periphery? And we found it's the sympathetic nervous system, the nervous system that controls parts of our stress response. And in fact, what is interesting that it innervates our bones. And in the bones, there is general formation of immune cells throughout our life. And these immune cells, in the presence of this, when the innervation to the bone marrow is altered, they are different. And then they are fighting the tumors much more effectively. So we can now manipulate them. So now one of the things that we are also doing, we are manipulating local innervations to tumors. For example, this is in the case of breast cancer. You can see here mice, you can see the tumors are much smaller when we denervate the innervation to the tumors. So our goal is eventually, by getting this understanding, to kind of merge the two fields, merging the mental state, the thoughts, and how they eventually change the innervation to the rest of the body and the physiology in order to boost the immune response and apply it in medicine. And in fact, we can do it by manipulating the brain with specific focused ultrasound or TMS, or one of the things that we are now doing in another center in Ikelov. We are actually, together with Professor Tarma Handler, we are using fMRI to teach people to activate these brain areas in order to boost their own response. And I would like basically to thank the people who are involved with that and all the funding, and you for all your support. And I think now we will be happy, Sarah, to answer your questions and any questions that emerge. Thank you.
Thank you, Asya. Adi, Irit, and Yuval, they have a script for me, but I need to go off script for a moment. I think everyone on this call has probably been touched by cancer in some way or knows someone who has. And it truly is, what can I say, not just illuminating, but so heartwarming that you have devoted your careers to really trying to make a difference for the people that we all know and love. It's exciting to see what you're doing. And I just am so hopeful that each and every one of you will be able to achieve the goals that you've set out today. Really, really, really tremendous. So before we open the floor to questions, I have a few of my own. First, is the cross-disciplinary structure of the RTICC helping to advance your research in ways that working at a more insular research facility would not? You all can choose who answers.
Avi, it's yours.
Cross-disciplinary. Yeah, so I would say, okay, I thought Yuval would take that one, but absolutely. I think that Technion really is about cross-disciplinary. It's about engineers and medical doctors and basic scientists working together. Absolutely, Sarah, that's what's unique about us. Having us look at the same problems that many other centers face, but we have a totally different set of tools at the Technion. And I think that's really what's unique. This is what's unique about this center, because there are cancer centers all around the world, but there's only one Technion Cancer Center that brings the best engineers, the best doctors, and the best basic scientists together. And I think that's really what's so special about it. And when you come,
it sounds like it's the Mayo Clinic of this area of research, and that you can all collaborate together.
I would say, Sarah, that Mayo Clinic is the Technion Cancer Center. But absolutely, there are colleagues, and they're amazing too. Yes.
So one more. Can you give us an idea of the path to commercialization for technologies and treatments resulting from research conducted at the RTICC?
I think I'll answer that. I think that one of the things that I showed in my presentation is that at the end of the day, it's not only about doing basic research. It's about trying to commercialize everything that we do in order to improve life of cancer patients. And we set as a goal at the RTICC to bring it to the clinic. We have at the Rambam Medical Center, we have one of the best in Israel of the phase 1 clinical studies, where the doctors there are part of the RTICC with pellets, for example. And we're working together in trying to translate everything that we do, either when we use nanomedicine that Avi Schroeder mentioned, or we use personalized medicine, as I mentioned. Each and one of us will try and work together with the clinicians in order to improve and to commercialize what we do, because then only this way we can bring it to the patients.
You know, if you can get to the bedside, the idea you pointed out of those little arrows getting straight through to the pancreatic cancer patients, wow. I mean, wow. So anyway, thank you. And now we'd like to open it up to all the viewers here for their questions. The panel will answer as many as they can. And if you have any questions, can you physically raise your hand? And Mary Ellen Tatani, the ATS Executive Director of Campaign Operations and Donor Relations, will unmute you. Mary Ellen, can you please take it from here?
Sure. Well, I'm just scanning the crowd here. I thought there was a question that came in early via email. I'll ask that and ask you to just raise your hand so I can find you later. So does any of what you have discussed today relate to any new treatments for lung cancer?
Irit, do you want to answer or should I answer?
I think that basically it's your research, so maybe you should answer. But yeah, lung cancer is a very tricky disease, and I think that most of what is being conducted today relates somehow to lung cancer. But Yuval, I think that you should answer that.
What I showed in my presentation is basically personalized medicine in lung cancer. So this is something that we're working on, and we are not the only one who's working on that. Advancing lung cancer in the last, I would say, decade is great due in part to the development of immunotherapy, immune checkpoint inhibitors, if you heard. And this is obviously one of the ways to improve lung cancer patients and to demonstrate or to display, at least display a benefit of this treatment. And I hope that with our technologies, we're able to predict those that will benefit from the treatment from those that do not. And those that do not, we can maybe suggest what kind of treatment they should get in order to respond to the to respond to treatment for lung cancer.
Thank you. Any other questions?
This is your chance, guys.
I think Sebastian has a question.
Yes, I do. I actually have a question for Professor Schroeder. To what extent will personalized medicine play a role in the future of cancer treatment? And how, if at all, does it inform your current research program?
Yeah, so thank you for the question. I think we're heading only to personalized medicine. We know today that even if we look at pain medication, one patient will respond to one medicine while another will respond to another. Unpersonalizing cancer medicine will be, I think, I don't want to sound dramatic, but it will be a crime in a couple of years because it'll be treating with a medicine that may be the wrong medicine for a patient when you know you can treat with a better medication. We're not there yet, but we're getting so much better. And I'll just give one example. So HER2, which is a specific breast cancer mutation, where in some cases, and actually also in some other cancers, also you'll find that mutation. And when you treat with a medicine that's targeted to HER2, it's a personalized medicine that gives you a response. And if that mutation doesn't exist, then there won't be any response or very little response. So we're already starting, but it's going to become huge over the next couple of years. It's happening in all the medical centers. Of course, at the Rambam Hospital, where Irit treats many patients on a daily manner. And I think we're in that era. We started, we're just opening the door. And as we look in, we see so many opportunities to improve the lives of so many patients. And I'll say that, as Sarah mentioned, that all of us were touched by cancer. No one, I think, today knows, unfortunately, doesn't know someone that wasn't touched by cancer. And I think when they come and when cancer patients come and approach us, scientists many times say, hold in there, try, do your best. Every day, new technologies are reaching the clinic, and many of them are personalized. Irit, you can probably give from the physician's aspect.
Yes, I would also like to say that this term personalized medicine is something that we take it into also the concept of, for example, the gender differences. If we go for the toxicity, for example, we know today that women and men, they don't, the toxicity profile is different. So, and mainly today, when we treat patients, we use only the weight and height, and that's it. And we don't take into consideration the gender. But maybe women should get a different protocol than men. This is something that is being explored now. And another aspect is the fact that we have huge databases that are being explored nowadays using tools of artificial intelligence and deep learning. So we can gather some analyzed conclusions and using these very high-tech technology tools in order to really portray the differences of what we assume is different. So it's not only that just the biology, but also trying to implement other tools as well of what we already have, because we gained huge databases of imaging, of all this clinical data, and it's there is a lot what to dig into.
Thank you all. Andy, did I see you with your hand up? I'm going to ask you to unmute. There we go.
So, okay, so I know that the TICC is on the campus of Rambam, correct? Okay. And I know that Technion is looking to build a human health initiative, which is going to be very similar for multidisciplinary, but on the campus of Technion. Is there going to be like a fast moving walkway between you? How are you going to integrate the cancer research with everything they're going to be doing at the Human Health Initiative? Anybody can answer that.
Can I? Yeah, so Andy, thank you so much for that question. I'll just take a first bite at it, but I just want to say that we're now in COVID times, and one of the projects that we're working on is with the Ramon Foundation, NASA, and high school students that want to run experiments at the Technion. And unfortunately, the Ministry of Education doesn't allow the students, the high school students, to enter the labs. And what we did, together with the help of the supporters of the Technion, Crown Foundation, which was mentioned right now, the Crown Vanguard, the Lewis family, and many other good friends of the Technion, we actually purchased a robot where now students can run experiments from afar. So you can sit at home, students actually can sit at home today and run an experiment in our labs at the Technion. You'll be able to sit in the United States and run an experiment in our labs. And when we started it, I wasn't sure what it would all be good for. And then a student entered my laboratory, and she was tears in her eyes, and she's a straight A student. And that I have to tell you, I was not a straight A student, okay? I was pretty far from that. These are like the super smart. She entered and I didn't understand why she's coming to our lab, and she said, listen, well, I have a problem. One of my hands doesn't work perfectly, and it's hard for me to pipette in the lab. And I said, well, probably that's why we purchased this robot, because you don't need to be in the lab, and we need your brains. That's what we really need. We need the smarts of you. So I think today the physical distance between the two campuses is becoming something that we can bridge. Listen, we're in some people's living rooms right now. It's not like shaking hands exactly, but it's not far from that, and we're communicating. So I think we're bridging it also at the research level and also at the technological level. Yuval?
I think you answered it really nicely, and in a way, I think that it all matters of collaboration, and the collaboration within centers is extremely important as well. I don't think that we need to compare or to compete or to do anything else. I think that these are two centers that will eventually will improve or benefit human health in general, I think the cancer center will focus mostly on cancer, and the others will do for other diseases and other welfare of humans. So I don't think that there is a problem here, and I think that it's all will eventually become as a united synergy project with everyone else. So,
if I may add, it also goes along with the trend now of what is called bioconvergence, which is the integration of engineering, computer science, and biology and medicine. And I think that the Human Health Initiative is really groundbreaking. It's something that is going to put the Technion and the TICC as the cancer part of the Technion in the top level worldwide, because this is something that is really very unique. It, for example, we have a common project, collaborative project between the TICC and Memorial Sloan Kettering and Rambam that really sought to use the data sciences and the biology expertise of the Technion. And as you probably know, MSK has a lot of expertise, but they asked to use this like the best part of the Technion and Rambam. And I think that this is an example for how an institution like that approaches us. And this is the best example. And the bioconvergence, which is now worldwide, I think that the Technion and the TICC would lead.
Okay, so we're going to take one more question. And for those of you that we're not getting to, apologies, but please send your questions to sebastian@ats.org, and we'll ask our esteemed Technion faculty members to answer them. I'm going to turn it over to Mary Sherman.
I just wanted to ask, since not everybody on the call is a scientific expert, for some clarification of the meaning of cytokine storm, since it's so important to the outcome of patients who have coronavirus.
Yuval, you want to answer? Or
I think that it's yours.
Okay. What the COVID does mainly in the lungs that generates the very severe infection in the lungs is due to the secretion of cytokines, secreted factors from the cells, and the virus induces the secretion of those factors, and this induces the inflammation, the severe inflammation. And it is called cytokine storm since it is like a huge acute infection and acute inflammation, something that is very, very, it's kind of like a storm. It's very, something that we see, and it's very fulminant, and it happens immediately. It can develop within days and hours. And that is why it is called the cytokine storm. And all those cases of severely infected COVID patients are characterized by this phenomenon.
And actually, I would like to add, interestingly, this cytokine storm kind of uncontrolled immune response, there are now studies showing that because it happens so fast, apparently there is, by regulating the vagus nerve, which is another system that goes from the brain, you can shut down this overwhelming immune response, and kind of it's a new strategy that might actually work to suppress this kind of reactions. And it's probably a system that is embedded in us as a way to regulate these kind of uncontrolled situations.
Asya, thank you. And Irit, Avi, Yuval, I mean, this really has just been fascinating. So a big, big thank you to all of you. Thank you to everyone who joined for being so supportive of Technion and allowing their research to happen so that we can find the types of cures that they're looking for. Everyone who is here is going to be emailed a link with a recording of this event, as well as an email address. And if you have any further questions, please don't hesitate to reach out to that email address. And this esteemed faculty and research group will be happy to get back to you. So thank you. I'm sorry that we ran a little late, but we thank you all for joining, and we hope that you enjoy the rest of the day. Bye-bye.
Thank you very much. Thank you.